Hello everyone, here we are going to discuss the morphological changes to differentiate reversible injury versus irreversible cell injury. So let's start it. So first I will let you know the introduction. Then I will let you know the diagrammatic presentation in which how we can differentiate the reversible cell injury from irreversible cell injury. We will see the two cells and we will see the diagrammatic presentation between them. Then we will see the nuclear features, the nuclear changes in irreversible cell injury, that is pycnosis, karyoraxis and karyolysis, the difference between them. And finally, we will see the differentiating table between the reversible and irreversible cell injury. So let's start it. So first, I told you that all cells remain in homeostasis. Human body is made up of cells. From head to toe, we are made up of millions of cells. All the cells are in homeostasis, that is they have a specific function and they have a specific structure. With their specific structure, they are performing their specific function. For example, the cells of salivary gland, they do the function of secretion. The cells of intestine, they do the function of absorption likewise. So they are in homeostasis. But whenever any stress, maybe physiological or pathological, whenever any stress is acting on the cell, then three things happen one by one. The first thing is cell adaptation. Cell will try to cope up the changes. The cell will try to cope up the stress and try to survive. So that is adaptation by doing certain structural changes inside that. There are 5 types of adaptation. I hope that you have watched my video on cell adaptation. So there is hypertrophy, hyperplasia, atrophy, metaplasia and dysplasia. These are the 5 types of adaptations. If the stress continues or stress is very severe, then the cell injury will take place. Initially reversible cell injury, and followed by, if the stress still continues, it will lead to irreversible cell injury. And between reversible and irreversible cell injury, there is a point of no return. So currently, I want you to tell the differences between reversible and irreversible cell injury. By the way, irreversible cell injury is equivalent to cell death. By irreversible cell injury, I mean cell death. So irreversible cell injury means the cell is already dead. There are 2 types of cell death that are discussed separately in different videos, apoptosis and necrosis. Watch that also. So how to differentiate the reversible cell injury from irreversible cell injury? Do you have, if I show you a cell, the diagram of a cell, looking at the diagram of the cell, can you identify, is it the cell showing reversible cell injury or is this cell showing irreversible cell injury? So what are the morphological differences between them? So we will discuss in the form of a diagram. We will compare 3 things. Can you see the 2 cells in front of you? In the diagram, you can see this is a cell showing reversible injury and this is a cell showing irreversible cell injury. So here we will compare 3 things. We will compare the cell membrane of both the cells. We will compare the organelles, 4 organelles in both the cells, and we will compare the nucleus in both the cells. So let's come on the cell membrane. Compare the cell membrane here with there. Can you compare? So in reversible cell injury, the cell membrane is intact. Can you see? Everywhere it's intact. Here the cell membrane is intact. Here the cell membrane is defective. In irreversible cell injury, the defects are there, the gaps are there, the defects are there. So cell membrane, there are defects. Here cell membrane is intact. Here cell membrane contains the defect. The first difference you can visible, it is seen in the diagram. Rather than learning, look at the diagram. The second thing, in reversible cell injury, blabs are present. Here the blabs are present. You can see in the cell membrane. Here blabs are absent. Here blabs are absent. There are no blabs here. Third difference. The third difference in the cell membrane. You can see here the cell is swelling. Overall the cell is swelling. Swelling is present. Over the cell is swelled out. The swelling, generalized swelling is there. Blabs is there. But here, since the defects are there, the extra cytoplasm will leak out. It will leak out. So there is no swelling. There is no swelling. So here the blabs are absent. Swelling is absent. So compare. So here cell membrane intact, here cell membrane is defective. Here blabs are present, here blabs are absent. Here swelling is present, here swelling is absent. So these are the 3 differences in the cell membrane. Reversible injury, irreversible injury. So in reversible injury, the cell membrane is intact. You can see blabs are present, you can see, and cells swelled out, you can see. In irreversible cell injury, the cell membrane is defective. The holes are present, the gaps are present, you can see. So extra cytoplasm leaks out. So no swelling, no blab. No swelling, no blab. So blab and swelling is a feature of reversible injury, not irreversible injury. So we have compared the cell membrane. Now coming on the 4 organelles. We will compare 4 organelles in each of them. So first compare the endoplasmic reticulum in both of them. Then compare the ribosomes in both of them. Here and there ribosomes. Then we compare the mitochondria in both of them. See the mitochondria in both of them. And finally compare the lysosomes in both of them. So compare the 4 organelles. Let's start with the endoplasmic reticulum. Here in reversible cell injury, the endoplasmic reticulum swells out. And here in irreversible cell injury, the endoplasmic reticulum ruptured. It is showing the lysis. So here endoplasmic reticulum shows swelling Here it shows lysis. Lysis means rupture. So that is the difference. Number two, the ribosomes are attached on the surface of the endoplasmic reticulum. You know, there are two types of endoplasmic reticulum, rough and smooth. The rough one contain the ribosomes on that, but ribosomes start dispersing. They move away. The ribosomes start dispersing from the endoplasmic reticulum, dispersion of the ribosome away from the endoplasmic reticulum. So rough endoplasmic reticulum is converting in the smooth one. So this is happening here also. It's not written, but you can write down dispersion of ribosome. So dispersion of ribosome is common in both. It is a common finding in both reversible as well as irreversible. The ribosome leave the endoplasmic reticulum. They disperse. They disperse. Now compare the mitochondria. See the mitochondria here. See the mitochondria here. Here also it is having the density, but densities are small. Here also we have the densities, but the densities are large and amorphous. Large and amorphous. So reversible cell injury, small densities in mitochondria. Irreversible cell injury, large amorphous densities in mitochondria. So that is the third organ. And the fourth organelle is the lysosome. Compare lysosomes in both of them. What is lysosome? Lysosome is the suicide organelle. Whenever it ruptures, the enzymes will come and the cell will do the suicide. So here the lysosome is eating its own organelle. Can you see a lysosome eating own mitochondria? It is known as autophagy. It is a suicide organelle. So lysosome is eating its own. It is eating its own organelle, that is showing autophagy and reversible injury. In irreversible cell injury, the lysosome also gets ruptured. So basically in irreversible, everything is getting ruptured. You can see the cell membrane is also having the defects. The endoplasmic reticulum is also having the lysis. The lysosome is also having the rupture or lysis. Okay, so that is the 4 cell organelles we have seen. So compare the 4 cell organelles, reversible injury and irreversible injury. Irreversible injury. What are the 4 organelles I told you? Compare endoplasmic reticulum in both of them, ribosome in both of them, mitochondria in both of them, and lysosomes in both of them. Can you do it? Can we do it together? Yes. So endoplasmic reticulum here, the endoplasmic reticulum swells. Here it shows lysis. Ribosome disperse. Here also disperse away from the endoplasmic reticulum. That is common in both. Mitochondria shows small density. Here it shows large density. Lysosome, here it is doing the autophagy. I mean, it is eating the own organelle, own mitochondria. Here it undergoes lysis or rupture. So please compare the 4 organelle. Compare the cell membrane. We have compared here. Compare the 4 organelle. Finally, compare the nucleus. Compare the nucleus and reversible and irreversible cell injury. Can you compare the nucleus, please? Can you please compare? Now see the nucleus here. Can you see the nucleus and reversible cell injury? The nucleus is showing only one thing, clumping of chromatin. The chromatin get clumped. So it's clumping of chromatin. The chromatin get clumping. Okay. What about irreversible cell injury? Here 3 things happen. Pycnosis, karyorrhexis, karyolysis. It is not one thing. 3 things happen one by one. Here only one thing, clumping is there. In reversible cell injury, only clumping is there. But in irreversible cell injury, 3 things happen one by one. That is pycnosis, karyorrhexis, karyolysis. So what do you mean by that? See the normal cell. In the normal cell, see the nucleus is blue in color, that is basophilic. And the cytoplasm is pink in color, that is eosinophilic. This is normal. This is normal. Now, during irreversible cell injury, first, the nucleus will get condensed. Condensation or shrinking. So condensation or shrinkage of the nucleus is known as pycnosis. Pycnosis is shrinkage or condensation. It becomes more blue because it got condensed. It becomes dark blue or more blue in color. The second, the nucleus will get fragmented into multiple pieces. Can you see? It get ruptured or fragmented into multiple fragments or pieces. It is known as fragmentation of the nucleus. The fragmentation of the nucleus is known as karyorrhexis. The fragmentation of the nucleus is known as karyorrhexis. Rhexis means fragmentation. And finally, the nucleus is losing its blue color. The blue color is lost. Can you see? It is loss of fading of basophilia. Loss of basophilia. It is known as karyolysis. Karyolysis is fading of basophilia. So I told you 3 terminologies: pycnosis, karyorrhexis, and karyolysis. Karyorrhexis and karyolysis. So what is pycnosis? Pycnosis is condensation or shrinkage of the nucleus. It becomes dark blue in color. Karyorrhexis is fragmentation of the nucleus. It gets fragmented into multiple pieces. And karyolysis is loss of basophilia or fading of basophilia. So blue color is lost. So one by one, these are the 3 things which occur in irreversible cell injury, not reversible. So compare the nucleus. Here in the nucleus, only one thing is happening. The chromatin get clumped. And here inside the nucleus, 3 things happen: pycnosis, karyorrhexis, and karyolysis. I hope you got it. I hope you got it. So compare the cell membrane in both of them. Compare the 4 organelles in both of them and compare the nucleus in both of them. Reversible versus irreversible. Compare the cell membrane. Here in reversible injury, this is reversible injury, this is irreversible injury. So in reversible cell injury, the cell is swelling. In irreversible injury, do not swell. Here in reversible, the cell membrane is intact. Here it disrupts. In reversible injury, the blebs are present. In irreversible, the blebs are absent. Compare the 4 organelles. Endoplasmic reticulum, here it is swelling, here it shows lysis. Ribosomes disperse in both of them. Mitochondria having swelling in both of them, having densities. Here densities are small, here densities are large. And lysosome, here it is showing autophagy, eating its own organelle. Here lysosomes get ruptured. What about the nucleus? Finally, nucleus. Here nucleus show one thing, here nucleus show 3 things. Please learn, compare the nucleus of both of them. We have compared the 4 cell organelles. Now compare the nucleus of both of them. So here nucleus show clumping. Here nucleus show pycnosis, karyolysis and karyolysis. I hope you got it. So please learn this difference table together. We can learn this difference table. Okay. So this is all about reversible versus irreversible injury. We know the introduction, that what is the sequence whenever a cell is in homeostasis. The first thing is cell adaptation, then reversible injury, then irreversible injury. Then in the diagram, show it rather than writing, show it in the diagram, and you can write the difference table. So in the difference table, we will try it once and we will revise it quickly. Reversible injury, irreversible injury. So in what points you have to differentiate it, you have to learn those points. Under what points you have to differentiate it. So differentiate it based on the cell membrane first. Then differentiate it based on the 4 organelles. Number 1, endoplasmic reticulum. Number 2, ribosomes. Number 3, mitochondria. And number 4, lysosomes. So compare under 4 organelles. And finally, lastly, tell me the nuclear features in both of them. So reversible versus irreversible. Here cell membrane is intact. Here it goes defects. It is having the defects. Here blebs are present. Here the blebs are absent. Here swelling is present. Here swelling is absent. So this is the differences between cell membrane of the two. Now coming on the differences between the organelles. Here endoplasmic reticulum swells. Here it go lysis. Ribosome get dispersed in both of them. Mitochondria here shows small density, here shows large density, mitochondria. And finally lysosomes. Here lysosomes undergo autophagy. Here lysosomes undergo rupturolysis. So that is the differences between the cell organelles between them. And finally the nucleus. Here nucleus show only one thing, clumping. Here nucleus show 3 things, pycnosis, karyoraxis, karyolysis. I hope we can do it in the exam, and rather than writing this, write this also and draw in your exam. If you are drawing it, you are getting more marks. Always remember, in university exam, draw more and more diagrams. That's all about it. Thank you. I hope you got it. So we can fill it, and I hope you all got it. Thank you.